This short video clip will hopefully give you a better intuition about what a principal component analysis, or a PCA, does to your data. We'll look at the two-dimensional case, this example here, because we can easily visualize that, but it does extend to higher dimensions as well. When we have a cloud of data points from our two variables, x1 and x2, we can fit a best fit line to the data that captures or summarizes as much of the information as possible, as much of the variation as possible. And that's represented by our green trend line here. This will be very close to a linear regression line, but it will be slightly different as we aren't trying to minimize the error terms only for x2. We'll fit a second line that is orthogonal or perpendicular to the first line, and again with the aim of capturing as much variation as possible. You can see that this blue line here captures the information about how the data are spread about the green line. In higher dimensions, we do this once for each variable in the data set, so we do this over and over again. We can then transform the data into its principal components. We don't lose any information, but we can summarize most of the information into fewer variables. This is called dimensionality reduction. We can see here that the first principal component from our data set captures most of the relevant information from the relationship between x1 and x2. So we could use this one variable in our analysis instead of both x1 and x2, while still capturing most of the information from our original data set. This type of dimensionality reduction, that is, reducing the number of dimensions of our data, can be very helpful when you're working with a large number of correlated variables. One practical application of principal component analysis is with genetic data. We can use a principal component analysis to capture broad trends of population substructure for our genetic data. This is a graphic from the Nature Genetics paper from 2016 by Price et al. I'll include the link to the paper below. People have been using PCA for looking at genetic data for a long time, but this was the first time someone suggested using the first few principal components to control for population substructure in a genome-wide study. Anyway, this figure displays the top two principal components from a PCA of European American samples. We see a large cluster here that they hypothesize represents the genetic variation in northwestern Europe. And then we have a smaller cluster along the first principal component representing genetic variation in southeast Europe, with the second principal component distinguishing between two Southeast European populations. These patterns can be captured with just a couple of principal components, or let's see, with just a couple principal components of a larger data set. We could summarize 1 million genetic markers, for example, in this way using principal components analysis. Or we can even get useful clusters like this with 100 carefully selected genetic markers. So in summary, principal component analysis, sometimes referred to as PCA, can be used to summarize information from many correlated variables into a few independent variables. This process is one way to reduce the dimensionality of our data. The resulting variables can be difficult to interpret, but they can be very useful for modeling trends in our data.